Hello friends, welcome back to my channel, and today in this video we are going to study about patterns of cytoplasmic inheritance. Now first of all, till now we learned about Mendel's principle of segregation and law of independent assortment. Now both the laws or these principles are valid on the genes that are located on chromosomes found in the nucleus. That is, it is for the majority of genetic characteristics because the majority of genetic characteristic depends upon the chromosome that is located inside the nucleus, where the Mendel's law of segregation as well as independent assortment works. So this assumption is valid for the genes that are located on chromosomes that is found in the nucleus. But in our body or in the body of multicellular organisms, we have some characteristics that is encoded by the genes which is located in the cytoplasm. That means apart from nucleus, we have genes also in the cytoplasm. And where in the cytoplasm exactly we have the genes? We have the genes inside the organelles, that is mitochondria in animal cells and chloroplast in plant cells. So these characteristics exhibit cytoplasmic inheritance, that is different from Mendel's principle of segregation and independent assortment. Or in other words, we can say the Mendel's principle do not work on the genes that is located in the cytoplasm. And inside the cytoplasm, where the genes are located? In the chloroplast and mitochondria. Now if see the mitochondria, the mitochondrion, that means a single mitochondria, consists of 15,000 nucleotides that encodes approximately 37 genes. Now, in a cell, we have 2 to 10 mitochondria, and each mitochondria consists of thousands of DNA. Now, what is the difference between nuclear inheritance and cytoplasmic inheritance? In case of nuclear inheritance, we already learned that the gene is transmitted to the next generation from both the parents. That is P1, P2, both the parents will donate their gamete and a zygote will form. So this zygote or the individual get the characteristics from both the parents. But in cytoplasmic inheritance, like in mitochondria and chloroplast, these organelles receive gene only from one gamete, which is mostly egg or the female parent or from mother. In some organism, it can be from male parent also or from both. But mostly, in most of the organism, the cytoplasmic organelle receive genes only from mother. This or such characteristics pass from mother to offspring and never pass from father to offspring. Now, these characteristics, they exhibit extensive phenotypic variations. Why so? Because there is no mitosis and meiosis, due to which there is no even distribution of genes. Now, as I told just now, we are multicellular organism. That means we have multiple cells in our body. Each cell consists of 2 to 10 mitochondria. If we talk about animal cells, each mitochondria again consists of thousands of DNA. Now, because there is no mitosis and meiosis and no even distribution of genes, therefore, say for example, if one cell is getting one mutated gene and one normal gene, any other cell can get two normal genes. The third cell can get two mutated genes because there is no even distribution of genes. So any cell can get any such combinations of genes. So due to which there exhibits extensive phenotypic variation. The same applies for chloroplast DNA as well. Now let's see this. Now this is one cell containing 4 mitochondria. Each is having 2 DNA. This is just for representation. Now after cell division, all the progenies can receive any combinations of such mitochondrial DNA. This white one, we can take it as a normal DNA. This red one is a mutated DNA. So because there is no even distribution of genes, therefore different cells will get different combinations of genes. So maybe it can happen that in one cell all 4 mutated mitochondria get transmitted. There can be a cell where 1 is normal, 3 is mutated. There can be where 3 are normal and this 1 is mutated. It can be 2 two normal, two mutated, it can be all normal. So we can see a range of phenotypic variations because of uneven distribution of genes. Now, cytoplasmic inheritance was first recognized by Karl Correns, and he proved that this is an exception to Mendel's principle. Because in case of cytoplasmic inheritance, the cytoplasmic gene do not follow the law of segregation and independent assortment. And on what plant he studied, he performed his experiment, that is on 4 o'clock plant, that is Mirabilis jalapa. And he observed the inheritance of leaf variegation. Now let's understand his experiment. What he did is, in case of this particular plant, he crossed pollen and egg, the male and the female part of a plant. Now, he found that when he crossed pollen obtained from white leaf and stemmed Mirabilis jalapa, the offsprings are all white. When he crossed between this white stem and leaf Mirabilis jalapa with the green one, that the normal one, the pollen obtained from the branch having the normal green leaf and stem. In that case also, all the offsprings are white. When again the same white stemmed and leaf Mirabilis jalapa egg is fused with variegated pollen, the pollen obtained from variegated plant. Variegation means, as you can see here, one leaf white, one leaf green. It can also happen that in the same leaf we'll get white and green patches. That is also a variegation or variations. So green means it is all normal chloroplast DNA. White means defective chloroplast DNA. So when it is all white, that means all the chloroplast DNA are defective. When it is all green, that means all the chloroplast DNA are green. When it is variegation, that means one white, one green, or the patches of white and green, that means it is a mixture of normal and defective chloroplast DNA. So he observed that when he fused the egg obtained from white Mirabilis jalapa and fused with pollen obtained from white Mirabilis jalapa, the offsprings are all white. But when female is from white Mirabilis jalapa and male part, that is pollen, is from normal green plant, in that case also all the offsprings are white. When the female from white and the male from variegated plant, then also all the offsprings are white. Next, when the female part is obtained from the normal green Mirabilis jalapa and fused with either of white, green or variegated plant's pollen, in that case, all the offsprings obtained was normal. So that means from these two set only, it is very much clear that the character is getting transferred only from the female part. The phenotype, if you can see the female, the same all the offsprings are getting. That means the genes are getting transferred only from the female part and not from the male part. Now, when the female part is obtained from variegated plant, in that case, what is the observation? Variegation in no particular ratio. That means because this female part is obtained from the plant which is variegated, that means variegated means it contains normal DNA also, normal chloroplast DNA also, and the mutated or the defective chloroplast DNA also. Because there is no mitosis or meiosis, so no even distribution of cell. So there can be cells where all the defected chloroplast DNA has passed in the next generation. So the offsprings will become all white. There can be the offsprings that are from all the normal chloroplast DNA. They will be green. There can be offsprings which obtained the combinations of normal and defective chloroplast DNA. So they will become variegated. And this ratio will also differ. Maybe in some plant, one leaf is normal, one leaf is variegated. There can be some plant where in one leaf only white and green patches will be observed. The ratio, the range of these patches can also differ. So what we can conclude with this, that is the phenotype of the progeny is determined by the phenotype of the branch from which the seed or egg is originated. That means the mother. It is only the mother who is responsible for the offspring genotype and phenotype because the genes are getting transferred only from the maternal side. Now, this is the experiment done on plant. Now, in humans also, a disease called Leber hereditary optic neuropathy, in which a rapid loss of vision in both the eyes happen. Now, this disease is a cytoplasmic inheritance disease. That means the disease is transferred from one generation to the next generation only through mother, or from mother only this particular gene is getting transferred because mother only donate the gene to the next generation because it is a cytoplasmic inheritance disease. Now, what will be the result of the cross when normal male is crossed with the diseased female? Diseased male is crossed with normal female and diseased male is crossed with diseased female. So you have to tell me what will be the result of the cross with respect to whether the offspring will become defective or the offspring will become normal. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.